On today's Arabidopsis Research Roundup, I'm joined by Sanjay Jian and Nick Cunniffe from the University of Cambridge, and they're going to be talking about a new paper just published in PLoS Pathogens. And the title is Virus Infection of Plants Alters Pollinator Preference, a Payback for Susceptible Hosts? ? So you might have seen some of this reporting on this paper in some of the popular press. It's been picked up in a number of outlets, but it's obviously good to talk to the authors involved with this. So guys, it'd be great if you could give an overview of this paper, what you found, and how you came to find it. That'd be great.
Quite interestingly, we found that the bumblebees are more attracted by the virus-infected plants. As we know, the bumblebees are pollinators. So in this case, it will help spread the pollen of susceptible plants. And then it's also beneficial to the virus as well, to help the virus population to maintain in the nature.
Okay. So intuitively, you might think that having a virus infecting a plant, it wouldn't be the best thing for a plant, but you found in a kind of bigger picture study that indeed there might be this trade-off between the virus infection and the plant's fertility. So can you discuss that a little bit?
Oh yeah, actually this finding was quite by accident. Our group leader, Dr. John Carr, wanted to explore the volatiles emitted by the virus-infected plants. But while he was waiting for the equipment, that was about 7 years ago.
Okay.
A collaborator who was Professor Beverly Glover, also head of the University of Cambridge Botanical Garden, offered him some bees to take a test.
Okay.
But surprisingly to us as well, the bees didn't go to the healthy plants. Rather, the bees went to the virus-infected plants.
Okay.
So that's quite interesting.
Okay, so this has got great significance for other studies about the evolutionary relationship between disease infection and disease susceptibility and the maintenance of genes within a population. And I know within the study you did a little modelling about that. So I don't know if Nick, if you've got some impact on that.
On the modelling side. So yeah, that was I'm Nick Cunniffe, and that was my interest in the study. I'm a mathematical modeller. So when John and Beverly described the finding to me, it was very exciting. It seemed exactly the opposite of what you'd expect a bee would do. So were I to be a bee, I'd probably want to go and pollinate a plant which wasn't virus infected, because it would be bigger and stronger. Of course, though, if plants which are infected are preferentially pollinated, that sets up the possibility of a trade-off. So what we haven't said thus far is that virus-infected plants set fewer seed.
Okay.
So they make smaller fruit and set fewer seeds. So all other things being equal, you'd expect virus susceptibility to be selected against, because those individuals make less progeny. But this mechanism with the pollinators being preferentially attracted can mean that that selective pressure doesn't get exerted because the susceptible plants are able to pass on their genes more readily to the next generation. Right. What the modelling component of the study did was wrap that up into a mathematical model and looked at two things. Firstly, whether the model parameterized or fixed to match the experimental conditions would be expected to lead in the long term to the susceptible plants surviving, and it did. But then with a model you can do lots of things, and one of the things you can do is to imagine lots of what-if scenarios. What if this was happening? What if that was happening? What if the parameters were this way? So we did a huge scan over many sets of parameters and found that the results were fairly generic. It isn't always the case that this mechanism would lead the genes to persist. In particular, if it were the case that virus-infected plants set no seed at all, they still could coexist with the plants which are non-susceptible because of the male side of reproduction. But if the model can get into the trade-off and can work out when one of three possibilities occurs, whether susceptible plants only dominate, whether resistant plants only dominate, or whether there's coexistence. And as you'd expect, if the costs of virus infection are low to moderate, and the benefits of virus infection in terms of the pollinators coming are moderate to high, then it tends to be that susceptible plants win out.
Right, okay. And what you found in this study, has it got implications? So I imagine it's got broad implications for other relationships in other systems. And are you looking at that now in plant system, or is there a need on this?
That would probably be the next step. As you probably know, the idea of maintenance of polymorphisms in plant populations do with our genes and things is incredibly well studied.
Yeah.
The next step for this study would start to think about adding the virus into the mix. In the mathematical model, we don't consider viral evolution.
Okay.
We only consider evolution of the host plant. So the next step would be to try and couple everything together.
Yeah, I see.
The modelling side, at least.
Okay. So let's just for the last couple of minutes, just go back to the early experiments. And, of course, GARNet's got a historic interest in Arabidopsis research. So, Sanjay, maybe you can talk a little bit about the work you did with Arabidopsis mutants to look at some gene expression change that you found differences between the ability of the viruses and their effect with the bumblebees.
Yeah, sure. Actually, we first used tomatoes as a model system. And then we found that the bees could be trained to tell the difference between the virus-infected plants and more inoculated plants.
Okay.
So then later on, we moved the model system to Arabidopsis.
Mm-hmm.
Although the bees do not naturally pollinate Arabidopsis flowers, but they could be trained to learn to tell the difference between the different Arabidopsis as well. Some of the Arabidopsis are virus infected, and some of them are healthy, non-infected. So, from the paper, you can see we got different learning curves. Like from the first choice and to the 100 choice, the bees could be trained to go to the different experimental setup.
Okay.
We use sucrose and quinine as different conditioning, so the bees could feel or smell the different volatiles emitted by different Arabidopsis. We used DCL1 and DCL24 and ACNO1, different Arabidopsis mutants as well. So from this, you can know these Arabidopsis mutants emit different volatiles, and the bees could perceive these volatiles.
Okay.
It's very interesting.
Yeah, so this nicely really links in viral infection with the kind of small RNA regulation, with the interaction with pollinators. So very much from the small to then the big picture. And I think that's one of the beauties that has been picked up on this paper. Although I guess you found a lot of the popular press pickup has been on the kind of the headline, viral infected plants are more susceptible. But really within this paper, there's some interesting stuff about gene expression. But then, as you talked about, some fundamentally interesting and new work about the relationship between susceptibility and disease resistance and things like that. So
exactly, yeah, it was very interesting. Well, firstly, gratifying how much the popular press picked it up. It was slightly disappointing that a few of the articles they seemed to think that the virus was being transmitted by the bees. Right. They kind of missed the point. But those who did understand what was going on, it was interesting to see how they span the work.
Yeah, absolutely, cool. Well, thanks very much, guys, for chatting to us today. And we'll certainly look to see if this paradigm is continued in plant science and in other systems as well. That'd be really interesting, I think. So, thanks so much.
Nice talking to you.